Now, primary databases. Primary nucleotide sequence repository. The main databases that store and provide the raw nucleic acid sequences are GenBank, EMBL and DDBJ. GenBank is located at USA and is accessible through NCBI portal. EMBL, that is European Molecular Biology Laboratory, is located at UK, and DDBJ, that is DNA Data Bank of Japan, is located at Japan. All the three have uniform data formats but not identical and exchange information in a daily basis. All these databases are the part of International Nucleotide Sequence Database collaboration. As all the three have same data format, GenBank database can be taken as an example to explain the data entry. As an example, we can discuss GenBank flat file format of Saccharomyces cerevisiae, that is yeast TCP1 beta gene. Please see the file details on the screen. Now, if we look at the structure in detail, if we take the field names in order, we can see the first entry as locus. Locus is nothing but the GenBank title that names the sequence entry. The accession number is given after the locus, which are unique identification numbers and can be used to retrieve information from the database by simply searching for the given accession numbers. The number of bases in the entry and nucleic acid type are also given after the accession number. Then, we can see a code called PRI code word used to classify the data. The last entry is the date on which the entry was made, and the next line is the definition that gives the definition of the entry with sequence name followed by the unique accession number. The version number shows that whether the entries have more than one version. Next item is the keyword that used to index the entries. Next is a set of source records that used to describe the organism from which sequence was it extracted. Complete scientific details are given followed by the publication details. Next is a citation section. Each entry is associated with a publication and is given in this section. The feature section includes coding region, exons, introns, promoters, alternate splicing mutation if present, and the translation into protein sequences if it code for one. Finally, the entry shows the sequence itself. The sequence is typed in lower cases and each line is divided into 2 6 columns of 10 bases each for simplicity. The number present before the sequence is the numbering of the bases. Primary protein sequence repositories. The two important protein sequence repositories are PIRPSD, that is Protein Information Resource Protein Sequence Database, at the National Biomedical Research Foundation, USA, and SwissProt at the Swiss Biotechnology Institute, Switzerland. PIRPSD. This is a collaborative endeavor between the PIR, the Munich Information Center for Protein Sequence, Germany, and the Japan International Protein Information Database, Japan. This is now improved as a comprehensive, non-redundant, expertly annotated, object-relational DBMS. The superfamily based classification of protein sequence is one of the unique characteristics of this database. PIR-PSD is available online at pir.georgetown.edu. SwissProt. This protein sequence database provides a high level of annotation and is available online at www.uniprot.org. The data entry can be considered as core data and annotated data. Core data is a sequence entered in single letter amino acid code, related references and bibliography. The taxonomy of the organism from which the sequence was obtained is also the part of this core data. The annotated data section contains the functions of the protein, post-translation modifications like phosphorylation, acetylation, etc., functional and structural domains and sites like calcium binding regions, zinc fingers, etc., and secondary structure features like alpha helix, beta sheet, etc., and similarities to other proteins if any, and diseases related with that particular protein. The entry in SwissProt and the explanation is shown in the table as on the screen. Example, platelet derived growth factor receptor alpha. The explanation for the codes used in SwissProt entry is given in the table we can see on the screen. First one is code, its expansion, and then the explanation. Code is ID, expansion is identification, explanation it gives the unique name for the entry and also the information about the entry. Then AC, it means accession number, This is the stable way of identifying the entry. The AC number will not change for any entry. If any entry has more than one AC, shows that the entry is generated by merging multiple entries. DT, it means date. We can see three different dates for an entry. Those are the creation date, the modification date of the sequence, and the annotation date respectively. DE, it's a description, gives a general description about the sequence. GN, it's a gene name, gives the name of the gene that code for the protein. OS is the organism name, the name and taxonomy of the organism. OG is the organelle, information regarding the organelle that contains the gene. OC, organism classification, information about the classification of the organism. RN, RP, RA, RX, RT and RL. All these refers to reference number, position, cross-reference, comments, authors, title and location. It explains bibliographic representation of the sequence, gives the information on the extent of work carried out by the authors. CC, it means comments, text document that contains extra information about the entry. DR, database cross-reference. It gives the cross-reference with other database which contain information regarding the same entry. KW means keywords, gives keywords that can be used as indices to find required information. FT means features table, describes the regions or sites of interest in the sequence, example, modifications, binding sites, etc. Last, SQ, it's a sequence header, indicates the beginning of the sequence data and gives a brief summary. Both PIR PSD and SwissProt use user-friendly software that helps a user to get only the required information. SwissProt also use a sequence retrieval system, that is SRS, that helps to search through other relevant databases like TREMBL for translated EMBL. TREMBL released as a supplement to SwissProt and is a computer annotated protein sequence database. It contains the translated sequence entry for all of the coding sequences present in EMBL nucleotide database, which was not fully annotated.